Hello guys, today our lecture is polynucleotides. We have already discussed the mononucleotide, dinucleotide. Today our lecture is polynucleotides. Guys, before start the lecture, we have a request that if you have not subscribe and like my channel, then please like, subscribe, and share my channels um, and lectures with your friends. Now start the lecture. Polynucleotides is at a voice from the name. Poly means many, nucleotide means nucleotides. So these nucleotides must contain the polynucleotides, mean mini nucleotides that are joined by um, ester bond, phosphate ester bond. Uh, First point is poly means many nucleotides, mini nucleotide. These type of nucleotides contain many nucleotide unit joined by glycosidic bond. Here I have write the glycosidic bond, but it is mistake. Here is the ester bond linkage to form polymer. It means that if many nucleotides are joined, we polymer means that if many substances are joined, which we call polymer. So here many nucleotides unit are joined to form polymer by the linkage of ester bond. Second point is there are two types of polynucleotides. We have two type of polynucleotide. We have already discussed in nucleic acids that nucleic acid is two type DNA and RNA. But nucleic acid is different from nucleotides in some um, terminology and some structure, because nucleic acid are polymer and nucleotide either be polymers um, or it may be two it may be two nucleotides, it may be one nucleotides, it may be mini nucleotide are called be nucleic acids. So DNA and RNA are the polynucleotide types, are the types of polynucleotide, and also DNA and RNA is called nucleic acid. Polynucleotide are also called nucleic acid. It is obviously the true. DNA means the abbreviation of DNA is deoxyribonucleotides. We will discuss in the DNA what does the deoxyribonucleotide means. RNA abbreviated is ribonucleotides. But here I only highlight the things deoxy. Deoxy mean the removal of oxygen. The removal of oxygen mean deoxy. Ribonucleotide, we have discussed that uh, nucleotides are made of three substances: ribose sugar, nitrogenous base, and phosphate. If the ribose sugar has 5 carbon. If from the number 2 carbon we remove the oxygen, if from number 2 carbon we remove the oxygen, then it will be converted to deoxy. It will be converted into deoxy. It means that DNA have no oxygen at carbon number 2. Carbon number 2 have OHM, the OHM, then we remove the oxygen from the OHM and hydrogen is remained. Hydrogen remain and oxygen is removed. Oxygen is removed and hydrogen remain, then we call then deoxy. Deoxy means removal of oxygen. RNA is ribonucleotide. RNA is uh, RNA RNA who no RNA who fully structure. Fully structure means that here we did not remove in oxygen from the ribose sugar. Mean normal structure of ribose sugar is called ribonucleotides, but it is a polymeric. But it is polymeric. uh, Here we discuss the next point is DNA. Uh, DNA made of deoxyribonucleotides. We have already discussed. It is mostly present in nucleus, but also mitochondria, plastid, etc. Here DNA have the location of nucleus. Mostly it is present in nucleus, but also mitochondria, plastid also contain their own DNA. But mostly DNA are present in nucleus. We will discuss the structure of DNA. Uh, Here we discuss the structure of DNA molecule in detail. In 1962, James Watson, Francis Crick, and Maurice Wilkins jointly received Nobel Prize in Physiology and Medicine for their 1953 determination of the structure of DNA. There are three scientists uh, that have started their function on the physiology and medicine, and also on the structure of DNA, and also on the structure of DNA in 1953. They determined the DNA structure. Jointly, they determined that DNA structure is by crystallography. By crystallography, they determined the structure of DNA, and they received Nobel Prize in 1962. They received Nobel Prize in 1962. They start their work on 1953, but they received Nobel Prize at 1962. According to these three scientists, according to him, DNA is double-stranded helical arrangement of strands around uh, one another. DNA, according to these scientists, DNA is double-stranded. Double-stranded mean they have two strands. Helical arrangement. These two strands are helically arrangement. These two strands have helical arrangement around one another. It means that they are arranged such a way that these two strands are in the helical form around one another. The two strands slide over one another, mean they are helical arrangement of these strands around one another. The base pair are between the base pair between the strand pointing of purine toward pyrimides. And these two strand one strand have purine pointing toward another strand of pyrimidines. Mean they are linked by purine and pyrimides. Mean these are nitrogenous bases. They are attached by these two strands. The helical strands are attached by purine and pyrimides between them, helping of molecule diameter at constant. These purine pointing toward pyrimides help the DNA constant diameter. DNA has diameter of 2 nanometer. To provide they help these purine and pyrimides bonding provide helping and diameter at constant 2 nanometer. They remain. They are able these two purine and pyrimides are able to constantly gives the DNA diameter at 2 nanometer. The base pair are flat with a distance of 0.34 nanometer between them. Um, Here is the point that the base pair, one in a single nucleotide in a single strand, one base is at a distance of 0.3 nanometer, 0.34 nanometer from another. Guys, this was the DNA structure and polynucleotide. In the next lecture, we will discuss details about the DNA structure and also some point related to DNA. Thank you for watching the video.